Okay, so it gives me great pleasure to introduce Professor Mark Schnitzer from Stanford. He's a HHMI investigator and a professor in Stanford's Departments of Applied Physics and Biology. His work has focused on the innovation and use of optical imaging technologies for understanding how large neural ensembles control animal behavior. His lab has invented several technologies now commercially available, including tiny microscopes that are small enough to be mounted on the head of a freely behaving mouse and that are now used by over 500 labs worldwide. So whenever you're ready, thanks.
Great. Thank you so much for the introduction and the invitation to be with you this afternoon. I'm going to share with you my lab's work on optical brain imaging as illustrated by this backdrop here in my title slide. And before I dive into the technical material, I thought it would be worth just saying a few words about the nature of our group's research. So our chief motivation is to understand how the brain processes information and to examine the computational basis for phenomena such as cognition and perception. As applied physicists, we often are pressed to develop new optical tools and approaches for the sake of addressing our questions about neural computation. And as I'll illustrate, many of these technologies produce large first-of-a-kind data sets, and one of our challenges is to develop computational pipelines that are up to the job of analyzing these large data sets. And overall, our intention and goal is to identify novel biological mechanisms underlying information processing in the brain. So there are several different types of activities here, ranging from the optical, the biological, the computational. But today, since this is an optics seminar, I'm going to largely focus on the optics and imaging side of our activities. So I'm going to try to illustrate the areas which have been a challenge traditionally in neuroscience, and in particular with regard to the use of microscopy for revealing brain dynamics. And I'm going to try to show you, largely by way of illustration, how we have tried to clear some of these challenges. And so this brief list here of impediments to imaging brain dynamics with light microscopy is one that I'm going to go through during the body of the talk in this sequential order. So first of all, I'm going to start off by discussing the fact that we would like to be able to image neurons and their dynamics, irrespective of whether they might lie near the brain surface or in deeper brain structures. Ideally, one would like to be able to track individual cells over many days in an animal's life, and this would allow researchers to look at neuronal changes during brain development or aging, or how neurons change their attributes during learning, long-term memory, or, for example, disease progression. Of course, we want to be able to image not just individual cells, but large populations of cells, and ideally we want to be able to do that in the same instance that we can observe what an animal's actually doing with regard to its behavior. So the goal here is to try to understand how neurons underlie animal behavior. Another challenge has been to watch multiple brain areas in action concurrently and to see what different populations of cells in distal regions of the brain may be doing to coordinate their activities and to drive animal behavior. Another challenge has concerned the fact that in the past, most of the light microscopy studies of neural dynamics have involved what are called fluorescent calcium reporters. And these give an optical report of a cell's intracellular concentration of calcium ions. And this has been useful because the intracellular concentration of calcium rises in response to neuronal spiking or action potentials. And so this has been a very useful approach for several decades now. The problem is that there is some loss of information as the neuronal membrane voltage dynamics is transduced into changes in calcium concentration, some loss of temporal resolution, and some lack of fidelity. And so one challenge in the field that's been a longstanding goal is to try to develop optical means of actually visualizing neuronal transmembrane potentials directly. And I'll show you some recent progress from our group and others in the field toward that goal. And this has allowed us to visualize in the brains of live animals neuronal spikes with millisecond and sub-millisecond time resolution. There are also very interesting subthreshold voltage dynamics in the brain, and these include brain waves or voltage oscillations and voltage waves that have long been known since the late 1920s, but have remained mysterious with regard to their mechanism. And then the final challenge I'll discuss near the end of the talk is how do we develop high throughput means of brain imaging? And here I mean largely in small animal species, and in particular I'll be focusing on fruit flies. Is it possible to increase the size of our data sets by developing technology allowing us to image many more animals than was previously feasible? So let's get started. I'm first going to discuss the challenge of imaging cells deep in brain tissue. And this slide illustrates some of this challenge. This is a sagittal view of the rat brain. And the chief impediment is actually light scattering, not light absorption at the wavelengths we typically use. And so for the typical confocal microscope, say confocal fluorescence microscopy, one can penetrate about 50 microns, maybe 100 microns at best into brain tissue, again, limited by light scattering. The two-photon fluorescence microscope can do substantially better and can image cells maybe 500, maybe 700 microns at best, deep into the tissue here as indicated by this green band. But there's a vast set of brain areas that lie out of reach of the conventional microscopy methods. And so the question is, how do we image these cells in these deep lying areas? And to do so with micro and scale resolution. And so an approach and a solution that we developed a number of years ago now when I was at Bell Labs is to directly insert optical microendoscopes into the brain tissue. And in some cases, these can be permanently implanted. You're seeing some examples here of these optical microendoscopes. This one here on the left is 1,000 microns in diameter. This one is 350 microns in diameter. These are gradient refractive index lenses, as you can probably infer from the flat face at the tip of each of these micro-optical objectives. And these little objective lenses are followed in each case by a longer but lower numerical aperture relay lens, whose main purpose is to give us the reach we need to penetrate directly into brain tissue. And these do provide the micron scale resolution we need to see cells. Nowadays, we have many different optical designs of these kind of probes. Here you can see some examples of doublets and triplet microendoscopes that we designed, all based on gradient refractive index microlenses. The objective lenses here are oriented to the left. The relays are coded black, and in the case of the triplets, we've added a third lens at the backside for numerical aperture matching. And here's an example of the refractive index profile of one of these lenses. I'm sure most of you are familiar with how gradient refractive index lenses work, and these are just some example profiles of lenses that we characterized when I was at Bell Labs. And you can see this one is on the tip of an optical fiber. Now, these lenses can be used in brain tissue in two main ways. We can use them with conventional epifluorescence or one-photon fluorescence imaging, as schematized on the left. And it's very similar to the conventional epifluorescence microscope, except that you've got this long, skinny probe, which is targeted to the brain area of interest. Or you can perform laser scanning two-photon microendoscopy, again with this probe, very similar in many regards to conventional laser scanning two-photon microscopy, but now the focal spot is relayed into the tissue through the microendoscope. And these two approaches have complementary strengths and limitations. The virtues of the epifluorescence microendoscopy is, first of all, its speed and simplicity, and the fact that you can acquire full frame images with a conventional camera at high frame rates. With the laser scanning two-photon microendoscopy, one has the virtue of optical sectioning, as in conventional two-photon microscopy, improved penetration depth into the tissue, and the ability to focally excite fluorescence at the laser spot, and to piece together images voxel by voxel. So we have one approach that offers speed and simplicity, and another that offers true three-dimensional optical sectioning. So that's how we image cells deep in the brain. And nowadays, our researchers are using this even in some of the most ventral portions of brain tissue, such as the hypothalamus, areas where people really had not visualized their cells routinely in live mammalian brains. Now, as I mentioned, we also want to be able to track individual cells over long periods of time, so we can watch what cells do or development during disease processes and so forth. And we have developed means by which either optical guide tubes or the microendoscopes themselves can be permanently implanted into brain tissue in live mice. And here you can see some example image panels of that. This is a transgenic mouse that is expressing a fluorescent protein in a subset of so-called hippocampal pyramidal cells. And if you look at these time-lapse image data sets, you can see that we can follow these individual neurons, in this case, using the 2-photon microendoscopy approach over periods of 2 months or more. And this middle set of images shows you a close-up of this little portion here. And you can see we can come back again and again, not only to the same individual cells, but in fact to the same dendritic processes. So we really have micron scale precision of return here in the brains of live mice. Now, as I'll show you in these next slides, we can even come back to individual synapses in the brains of adult mice. This was something that was pioneered by Alessio Ottardo and James Fitzgerald in the lab. Alessio now has his own group in Munich, and James has his own group at HHMI Julia Farm in Ashburn, Virginia. And dendritic spines are the postsynaptic morphological structures at a large majority of excitatory synapses in the brain. And here you can see in these same transgenic mice in which there was expressed a fluorescent protein, that with high resolution microendoscopes, Alessio and James could actually visualize these postsynaptic structures. These micron scale knobby structures represent the morphological elements at in the postsynaptic cell. And whereas the dendrites were stable over long periods of time in the adult brain, we found that in this case, the synapses were not stable, and we could visualize some of these dendritic spines that appeared midway through a time-lapse experiment, others that disappeared, as indicated by these different colors of arrowheads. And so this general approach has given us new opportunities to track how the brain's connectivity, the connections between neurons, may change in response to different life events. And we can do that in the live adult brain down here at the synaptic scale. So this allowed us to ask the question of whether or not in the hippocampus, which has been thought to be an organ of memory retention, the connectivity patterns, as visualized here by way of the spines, might exhibit turnover kinetics that matched the established longevity of memory retention. And one of the outcomes of this story, which was published about 5 years ago, was indeed that there was this correspondence between behavioral testing of memory and the microscopic kinetics that we were seeing of the synaptic elements. So the two were broadly consistent, as if the connections were what was storing the memory information. Now, another important challenge that we've had to deal with in the field is the question of how do we visualize neurons and their dynamics, not just in live animals, but in animals that are non-anesthetized and free to perform interesting behaviors in the lab. And we had devoted several years of work to the construction of miniature microscopes that were small and light enough that they could be mounted on the head of an adult mouse. We had been doing this with fiber optic systems. At some point, we appreciated that cell phone CMOS cameras and CMOS image sensor chips had become sufficiently good in their properties that we should really be considering making an integrated miniature microscope. And these cameras, of course, we all nowadays carry about in our pockets and our cell phones. And so we set about building an integrated miniature system with all the optical components contained in the miniature microscope with no fiber optics running to remote light sources or remote detectors. So this shows you a photograph of one of the miniature microscopes we built at Stanford. It's 2 grams in mass, shown here on the tip of a finger, based on semiconductor optoelectronics. And you can see various elements. This is the little micro-optical objective lens. There's a PCB, a printed circuit board here on the side where there's LED illumination placed. There's miniaturized fluorescence and dichroic set here, and there's a cell phone CMOS camera here, and the lines are bringing the data off are strictly electronic, so they're floppy mechanically and allow the mouse to behave in a natural manner. Now, I should also say by way of disclosure that my colleagues and I took this technology at Stanford and licensed it from a startup company that's spun out almost a decade ago now. And subsequently, this technology through the company and Scopex has made the approach commercially available. Over 400 labs are using it now. And I continue to consult for the company and have a financial interest. And so that's a disclosure for you. Now this slide shows you a little bit more about the elements of the microscope, as well as those that did the work. So Kunal Ghosh was an electrical engineering student at Stanford who was jointly advised by myself and Abbas Al-Gamal. Abbas was one of the pioneers in CMOS image sensor chips. Laurie Burns, an applied physicist who did the optical design. Eric Cocker, a mechanical engineer who did the mechanical design of the microscope and this focusing screw cap that you can see here. So this was fodder for 3 different PhD theses. So here you can see a schematic of how the instrument is used. One selects a microendoscope appropriate to the brain area that you want to target. That is then implanted into the tissue. And then the microscope, shown here, can be taken on and off different animals so that you can use one microscope on multiple different mice. And over here we see some photographs of the original device. Again, you can see the micro-optical objective, the PCB with the LED source here on the side, cell phone CMOS camera on the top, miniaturized dichroic and filter source there. Now this video shows you an example of a mouse wearing the microscope. They can behave quite naturally. They can run it at full speeds. And so the approach is generally applicable to most of the behavioral assays that are widely used in neuroscience. Now, while the animal's wearing the system, we can then visualize the dynamics of large sets of neurons. So this is illustrated here. This is work of Jones Parker, Biafra Hananu, and Jesse Marshall. Jesse's now at Harvard, Biafra is at UCSF, and Jones has his own group at Northwestern. And in this study, they targeted the microendoscope and the microscope to look at so-called medium spiny neurons in the dorsal striatum. And this slide gives you a sense of what a good afternoon can look like in the lab when you're using this technology. You can have multiple mice plugged in, and you're acquiring digital imaging data, and you can be tracking hundreds of neurons per animal. And you can do this afternoon to really see what individual cells may be doing over long periods of time in conjunction with tracking animal behavior. So I think you can get a sense now of why we also place a lot of emphasis on the computational analysis of our data sets, because we're easily getting multiple terabytes from these biological studies. Now I'm going to give you a little bit of familiarity with what one can do with these data sets. So to illustrate this, I'm going to show you some data that we took in the hippocampus. This is work done by Yaniv Ziv and Laurie Burns. Yaniv has his own group now at the Weizmann Institute. And so we targeted, in this study, the microendoscope to the hippocampus, illustrated here. And this was a long-term study. And we were looking at hippocampal representations of space. So the animals were behaving naturally within various enclosures. Here you can see an animal within a 2-dimensional circular arena. And meanwhile, on the right, you can see the calcium activity of hippocampal neurons. And these neurons were engineered to express a so-called fluorescence indicator of the calcium concentration. And as I mentioned earlier, this intracellular calcium concentration level is a downstream indicator of somatic or action potentials or cell body firing. And so we're looking at an indirect measure of that using this so-called genetically encoded calcium indicator. So it's a fluorescent protein that modulates its intensity based on the calcium concentration level. Now, what's interesting about this set of neurons is that they are responsive when the animal is located in different portions of space, in different portions of its arena. So we can take the data sets and we can analyze them. And so here's an example from one mouse in which we computationally extracted over 700 individual neurons from the type of movie that I just showed you. Their cell bodies are marked here in red at the top of the green fluorescence background. You can see the a mouse up close wearing this. In some of our more recent data sets, we can even track over 1,000 cells. This is one of our best mice ever, 1,202. Now, what I want to show you is what we can get from the data when we examine a little bit more closely the activity patterns of those individual cells. And it had been known, including from a lot of work done at Arizona, that these neurons were responsive to different locations in space. So they had preferences when the animal was located in different locations in the arena. And this is a lot of work has been done at this in Arizona by researchers such as Carl Barnes and Bruce McNaughton when he was there. And here you can see some examples of this, but not determined electrophysiologically, as was classically done but optically. And so these are bird's eye views of the kind of arena I just showed you, a circular arena, where we can pick up the mouse and move it to a square box. And here you can see that the different cells have different preferences. So for example, cell 348 tends to fire here in the northeast of the circular box, but if you pick up the mouse and move it to the square box, you can see that this cell prefers to fire when the animal's in the south. And not every cell is active in every arena. So this cell 159 is active in the northwest of the square box, not so much in the circular box. And here cell 198 is active in the southwest, but not so much in this square box. And so this kind of phenomena had been documented previously electrophysiologically, but what was new is that because we had our long-term imaging capacity, we could see how these maps of space actually might evolve. And so I'm going to show you this a little bit in more detail. So this is a mouse running the world's simplest maze, the linear track. And what I want you to do is focus on that cell there that's been circled and look where the mouse is when that cell is active. And what you'll see is that there's a particular location in space that we've marked with this arrow when the mouse acts to occupy for that cell to be active. And you'll see, moreover, that the animal has got to be going from top to bottom, not bottom to top. And so when you see that cell fire, you know not only where the mouse is, but also in which direction it's headed. And so therefore, by extension, you might imagine that if we could track the activity of all the cells in review, you might in fact be able to predict or reconstruct the animal's entire passage back and forth in the track just by looking at the brain video. And so that's exactly what Lacey Kitch did. So the black traces here, back and forth, show the animal's actual passage through space. And the red is the predicted movement of the mouse that you would infer. She built a decoder using a portion of the data to train the decoder and then test it on the remaining data. And so here you can see that just based on the brain activity, one can predict quite accurately where the mouse is in space. Now, what was interesting about this is that we could ask whether or not these maps of space were stable over time. So here, Lacey performed what we call time-lapse decoding to see how permanent or transient these memories might be. So in this case, she trained the decoder on data from one day, and then subsequently in the experiment, the animal was placed back in the track, get a new data set, and then we could see whether or not that decoder was successful at predicting the animal's passage with this 5-day interval. Or we could use a greater interval, 15 days here in the middle data, or as long an interval as 30 days. And what you can see is that there is a decline in the accuracy of decoding. But nevertheless, even with a 30-day interval between training and testing of the decoder, the estimation is highly significant, indicating that there is a long-term representation of that track in the mouse's brain that is still faithful to a high degree over this long-term time period. And so this gives you a sense of how we can use these data sets to look at substrates of memory and representations in large populations of cells using our optical technology. And in more recent studies, we've been looking not just at simple linear tracks, but at more complicated mazes and trying to understand from our tracking of neuronal activation patterns how animals may learn the maps of new environments they haven't encountered before and then recall them subsequently on later days of the experiment. Now I'm going to give you a flavor for some of the more recent developments with the miniature microscopes. So the original version was for imaging only. Inscopics now has a new version which allows 3D focusing, electronic focusing, as well as optogenetics. So in other words, optical manipulations of cellular activity at the same field of view in which we were performing imaging. Another important development has been the expansion of the species to which this technology can be applied. So we had initially focused on mice, one of the primary model species used in neuroscience for the mammalian brain. But more recently, there has been increased attention on the primate brain due to the hope that primates will provide a genetic model species that may allow us to study human brain disease in ways that have not previously been feasible. And in Japan, the marmoset has been a major focus of the Japan Brain Minds project. And we collaborated with Hideyuki Okano's group in Tokyo. The work was all done there, and I had a minor role in this. And the Okano lab was able to very quickly adopt the miniature microscope technology to the marmosets. And here I'm going to show you an example from Keio University in Tokyo. You can see the animals wearing the microscope. And the animal is being given food pellets of reward. And meanwhile, you can track activity here in the motor cortex. And you can see some of the cells, which are marked there with different colored circles, that are active when the animal reaches to the left, the right, or in both directions. Here's another example. In this case, the animal's wearing the microscope and is climbing a ladder, and the cells that the O'Connor group has circled in green indicate those that are active when the right forearm is reaching for the next rung of the ladder. And they are very active now in developing models of human brain diseases and using the microscope to take a look inside the marmoset brain to see what goes awry in various brain conditions. Okay. Now, so far, everything I've told you about has concerned populations of cells at a single locus of brain imaging. And one thing we would be very interested towards understanding is how do we go beyond this level? So we've been looking, everything I've shown you so far has been so-called neural populations or neural ensembles. But ideally, we'd like to be able to understand how ensembles of cells in 2 or more brain areas work together. So multi-area networks of cells, how do they control animal behavior? And for this, we need to consider microscopes that may have different formats than the conventional microscopy format. And so I'm going to illustrate what I mean by this. So one very simple way of trying to image neuronal activity in more than one brain area at a time is to make use of the small size of the microendoscopes. And so very simply, 2 or more microendoscopes can be implanted into the same brain. And this should allow one to look at brain area interactions. So for example, you can see here with this CT scan based model of the mouse brain that there's quite a lot of space actually to pack these microendoscopes in closely and to interrogate multiple brain areas concurrently. So here's an example of what one can do with careful surgical planning. So here's a mouse that is wearing 2 of these miniature microscopes. You can probably just see them by virtue of the small LED illumination. There, obviously have a bird's eye view of the mouse. And one microscope is targeting the hippocampus, as shown by the video on the bottom, and the other is targeting the prefrontal cortex. And meanwhile, the mouse is running this simple plus maze, and we can try to understand how these 2 brain areas may guide the mouse in its choices of which way to go. So that's a case in which the animal is freely behaving. Now, we can also use a complementary approach in which the animal is held in place on a more conventional looking apparatus, but is nevertheless free to behave in place. And this was an approach that was originally developed in our lab by Jerome Lecoq. He is now a senior scientist at the Allen Institute in Seattle. And with his collaborators in the group, he developed a system we call the duopus. This is a double-armed 2-photon microscope. And you can see here in the schematic that the system is based on a single titanium sapphire laser that is split into 2 optical pathways. Each one is sort of a microscope in its own right, equipped with its own photodetector. And each one of these pathways terminates in a microendoscope. And because the system is mechanically articulated, it can be directed to the brain area of your choice, allowing you to image 2 brain areas at once. So here's an example. This is a collaboration with my colleague Lixian Lo, and Tony and Mark Wagner took the lead on this in the lab. Here you can see an animal that has learned to do an arm pulling assay. And so you can see the mouse is holding this little handle of this robotic manipulandum, which allows us to track the animal's motor behavior. And meanwhile, we're imaging 2 brain areas that are intimately involved in motor control. First, we're imaging the cerebellum, and secondly, we're imaging activity in the motor cortex. Now, what's interesting about this is that these cells are only 2 synapses away, but anatomically they're quite far. And so we needed to have a flexible, mechanically articulated system to be able to access these 2 distal areas that are nevertheless working in tandem. So just a couple more remarks about this approach. So because of its mechanical flexibility, the duopus can inspect areas that are either nearby or further away. So here's some examples. So these are pairs of images acquired simultaneously in the M1 motor cortical area as well as the somatosensory area S1, schematized here from a bird's eye view of the mouse brain. Or you can target, for example, 2 different visual cortical areas, V1 and LM. Now, because these are microendoscopes, you're not limited to superficial areas. Of course, you can go in image deep areas as well. So here's an example of a pair of images that was acquired in the CA1 portion of hippocampus, as well as a frontal association area in the neocortex. And so one important takeaway from this portion of the talk is that this double-armed microscope can image pairs of brain areas that are inaccessible through large cranial windows. So many labs in the field have been applying large cover slips, essentially, or large cover glasses to permanently encase the brain but provide optical access. But this limits you to sets of areas that are anatomically contiguous. And here we're looking at areas that are not superficial and need not be contiguous. We have a complementary capability here due to the mechanical flexibility in the engineering design. Now, another system that does provide a large field of view is this one. This was published recently this year and was led by applied physicists Oleg Rumyantsev, Jerome Lecoq, and robotic engineer Joan Saval. This is a system that uses a large objective. It's sort of a 2-photon mesoscope, as it's sometimes called in the field. And what we do is we take the infrared ultra-short pulse beam from one sapphire laser. Then using a set of 3 electro-optic modulators, the beam is split between 16 different pathways arranged in a 4x4 checkerboard pattern at the focal plane. And here you can see the schematic of these beams running parallel to the optical table. They are brought to a focus on, they're brought here on one of the galvanometers, and there's another galvanometer, and then these simultaneously scan all 16 spots of light here, all 16 foci in the animal's brain. Now, one facet of this is that not all 16 beams are activated at the same time. So this is due to the nonlinearity of 2-photon excitation. It's much more efficient to activate only portions of the beams. In reality, we activate typically only 4 at a time. Each of the pathways has a corresponding detector, as schematized here. And this shows you a little bit more about the pattern of spatial-temporal multiplexing. So when we activate the beams, we're usually doing it in 4 sets of 4, as schematized here. You can see, say, 1 set of 4 indicated by the Roman numeral 1, 2s, and so forth. And this is not only a more efficient manner of excitation, but also allows us to computationally correct for fluorescence scattering from one image tile into adjacent image tiles in a way that I'll illustrate here. So here you can see on the left some of the timing protocols. This illustrates the 4 different phases of the acquisition of the fluorescence photons for 1 pixel. And of course, 4 beams are on. And you can see examples of the raw data here. So this is, say, what we would acquire from the 16 PMTs during phase 2, phase 3, and phase 4. But there are scattered photons, fluorescence photons, that might be excited in one image tile and that would scatter to one of the other detectors. And the fact that each activated tile is surrounded by tiles in which the laser is inactivated allows us to make some good inferences about which tile a photon likely originated from. And that is indicated here in these colored patterns. So for example, if the excitation laser is on one side of an activated tile and you catch that photon, say here, it is much more likely to have come from this tile than from this one, just due to the distances involved. So in that way, we can correct for inter-tile scattering. I'm going to show you the effects of this. So here's an example image after we've put together the images from those 4 different phases, but you can see the effects of scattering at the edges between the tiles. And now here is after the unmixing, after the correction. So this is an important aspect of this multi-tile imaging approach. We've also shown that after the correction, there's very little residual effect on our ability to sample the calcium activity traces of the neurons. And one can test this very simply. In one configuration, you would have the beam from only one tile on, so there's no crosstalk coming in from adjacent tiles. And another configuration, you could have only the crosstalk, so you've left that laser beam off, you can measure the crosstalk, and you can check your ability to detect cells and their activity patterns with or without crosstalk. And in this way, we showed there was essentially no effect after the corrections. So I'm going to show you some example images. I want to just mention important collaborators on this. So in addition to Oleg and Jerome, we had the benefit of a collaboration with Honghui Zhang, who's now director of the Allen Institute. And we used triple transgenic mice that she and her team made. These animals were expressing a particular genetically encoded calcium indicator known as GCAMP6F in a subset of cortical pyramidal cells, the so-called layer 2-3 pyramidal cells. And you can see an example image taken with a 16-beam microscope. The neurons here are highlighted in green, and you can see the cells atop the background fluorescence. And these stripes here are the blood vessels in the cortex. So here's an example movie. You can probably just make out the seams between the 16 tiles. The twinkling that you're seeing, that's neuronal activation. So each one of those little dots is a cell body of one of these layered 2-3 pyramidal cells. And so in this way, we can track an area within the field of view that is 4 millimeters squared, so 2 millimeters on a side. And that is sufficiently large that we can track neurons, say, across the entire primary visual cortex of the awake adult mouse. And that has, in turn, allowed us to ask some new questions about visual cortical coding. Now, here's another example. And this was a little bit later in our studies, and the stitching between the tiles was better. And so you may have a harder time seeing the boundaries between the different image tiles here. And additionally, you can also see some dendritic activation. So whereas in the previous movie, you could only see the cell bodies, now you can see some of the proximal dendrites activating when the cells are firing in response to a visual stimulus that we're showing the mouse on a video monitor as it sits underneath the objective lens. Now, from that previous movie, it was probably hard for you to infer what the mouse was actually watching, but in some areas you can actually infer. So this is a movie from the superior colliculus, and we're showing a moving bar on the video monitor. And in turn, you can see a moving bar type of activation here in the superior colliculus in response. So this really shows you that there is a so-called retinotopic map in some of these visual structures that provides a topographic representation of what's taking place in the external visual world. And so I'm not going to go into the details of what we did in this biological study, but the breadth of this microscope's field of view, allowing us to cover the entire primary visual cortex in the adult mouse, allowed us to take a look at the fidelity with which visual scenes could be represented, and to actually take a look at what might be some fundamental constraints on the accuracy of that code. And so that study appeared recently and was a close collaboration with theoretical neuroscientist Suri Ganguly at Stanford. Now, we of course would like to image multiple areas, many areas at once. And I'm going to show you an animal preparation that has been very useful for that. This was work of Tony Kim and Yanping Zhang in the lab. And they developed a fairly simple preparation. This is a curved glass optical window whose radius of curvature matches the natural curvature of the mouse cranium. And with a careful surgery, it can be permanently implanted to replace the bone, thereby giving you chronic optical access to the brain and the cells and the processes. And if this is done well, you see no indication, even with cellular resolution, of inflammatory processes. So here you can see we've been looking at astrocytic activation, comparing non-surgical control to tissue that had received the curved glass window, no indication of glial activation. So this can be done successfully. And I'm going to show you what this looks like. In fact, the data is the same as that behind me in the backdrop here in Zoom. This is a running mouse. The animal is fixed under the objective lens of a large macroscope. The animal is similar to what I had been describing previously, in which there's a genetically encoded calcium indicator in the layer 2-3 pyramidal cells. So especially on the midline, you can probably see the activation of those neurons. The brain is bowing out of the field of view of this microscope, so you see it gets a little bit fuzzier here on the periphery. But nevertheless, we're looking at on the order of, say, 40 cortical areas at once. And this is chronic in the sense that you can come back again and again to the same animal over the course of an extended behavioral protocol for many days. So now I'm going to zoom in a little bit and show you some more of the details that you can see in this preparation. So here At higher magnification, I think you can more closely see the activation of the individual neurons. With this curved glass window, you can also see some of the hemodynamics that are taking place in the vascular constrictions, especially on the, if you look at the midline vessel, you can make some inferences about what the animal is running. And if I zoom in even further, now you can even more clearly see the activity of the individual cells. So this panel here is from this boxed region over here, and you can very clearly see the individual cell bodies as well as the blood flow through these cerebral vessels. Okay, so this is a very useful tool for allowing us to look at multi-area interactions. When we have compared the data to publicly available maps of the mouse brain, such as the Allen brain map, we find that we can sample cells across 36 to 40 areas in individual preparations from frontal to occipital lobes. And one can apply different optical modalities to these animals, 2 photon imaging, 1 photon imaging. So here you can see some example data sets. These are montages. So here, for example, in panel B, we're looking at a montage in the coronal plane, and down here in E, and from this portion here in the sagittal plane, we can see these pyramidal cells, both in the cell body layer, as indicated here, dendrites, and we can even get down to the level of dendritic spines, as I showed you earlier, at the level of individual synapses. So this is a terrific new preparation that we are now actively making use of to study a variety of processes. Now, one important area of progress that I mentioned at the very beginning was that up until this point in the talk, everything I've been showing you with regard to brain activity has been using these fluorescent calcium indicators, which a low-pass filter spiking activity to a certain extent, and which also omit certain facets of membrane potential dynamics, and in particular the so-called subthreshold dynamics, the voltage activity below the spike threshold. And so there's been a long-standing push in the field to develop optical means of tracking neuronal membrane voltage directly. And this goes back several decades to researchers who developed what were called potentiometric dyes or voltage-sensitive dyes. These were pioneering but tended to be quite phototoxic and to bleach rapidly. In more recent years, there's been an effort to develop genetically encoded voltage indicators, and that's where our group and others more recently have been focusing their attention. And of course, what we'd love to do is be able to combine these kind of fluorescent protein indicators of neuronal voltage with the optical hardware that I've been describing. So I'm going to show you some examples of optical readout of voltage activity. Our work in this regard was initiated by Yi Gong, who now has his own group at Duke University. And here you can see an example cell, an example neuron in vitro that he recorded electrically using a patch electrode, and that trace is shown here in black, as well as optically using a particular type of genetically encoded voltage indicator that makes use of FRET, fluorescence resonance energy transfer, and an opsin protein. So we call these indicators FRET opsins. And what this slide illustrates is that the optical readout provides a very faithful record of not only the cell's spikes, but also various facets of the subthreshold activity. So you can see the form of the action potential and the rise to the action potential. And this particular indicator was, we called it ACE2NM neon green, or ACEM neon for short. And it uses a very bright genetically encoded fluorescent protein. M neon green is even brighter than GFP, coupled to a voltage sensitive opsin. And with the milliwatts of light, we can track spiking events and subthreshold events like this. So I'm going to show you a little bit about how this works. As I said, this indicator is a fusion of 2 different protein moieties, a opsin protein, which has voltage sensitive light absorption, and there's a FRET interaction with this bright genetically encoded fluorophore. So one advantage of this FRET design is the fact that we can decouple the voltage sensitive portion from the portion that gives us the optical signal. And so when we're doing protein engineering, we don't have to worry about disrupting one when we adjust the other. So the proteins in the opsin family were previously established by people such as Adam Cohen at Harvard to have voltage sensitive fluorescence. And this is due in large part due to voltage sensitivity of the optical absorption. And this is known to occur through a proton protonation deprotonation event that depends on the membrane voltage. So these are membrane bound proteins, and when the membrane voltage changes, there's movement of this protein, which in turn changes the absorption spectrum. So we can take advantage of this, and one can use these protons, these proteins directly as voltage indicators in their own right. That has the disadvantages that their fluorescence is rather dim, and it requires actually large illumination intensities, which run the risk of being damaging to the brain. So instead, we've coupled these through a FRET reaction to a very bright fluorescent protein, and thereby can look at the efficiency of the FRET as a function of the membrane voltage. And so this slide shows you that this strategy works very well in live mice. Here you can see a neuron that we've labeled with this green voltage indicator. And we're also targeting that same cell with a pipette here, an electrical recording pipette filled with a red dye. And so here in the black traces, we have a record of the cell's spikes as acquired electrically. And here in the blue traces, you can see a report of the cell's spikes from the optical measurement. And we catch each and every spike in this way. And moreover, we can time the spikes very accurately. So when we use the electrical traces as the ground truth, and we estimate from the optical trace the occurrence time of the spike, you can see that the distribution of the errors is nicely Gaussian with a standard deviation of about 200 microseconds. So we get exquisite timing precision with this approach. And of course, because it's genetically targeted, we can direct the indicator to neurons whose type we can select. Okay, so this shows you the function of these indicators in the mouse brain. We've done this in other species. This is data from the fly brain, data taken by Chung Huang in the lab. Again, using modest illumination intensities, unlike those used with the opsins. Here you can see, again, in this dual electrical optical recording, we're catching each and every spike as well as these subthreshold depolarizations. And here's a magnified view of that. And again, as in the mouse, in the fly, we get highly precise timing of the action potentials to sub-millisecond precision levels. Now, recently we've developed with collaborators the use of different colors of these FRET opsin indicators. So in work led by Madhu Kannan and Ganesh Vasan in Vince Peerbone's lab, they used this FRET opsin principle, but substituted another genetically encoded fluorescent protein for the ASEM-Neon. They used an mRuby, a red fluorophore, to create a red fluorescent voltage indicator that they called Varnum. And because you now have two different colors of FRET opsin indicators, there's the possibility of tracking two different genetically targeted neuron types at once in a live animal, and that is something we have done. So this is data from Chung Huang here in his flies, which you can see under the objective lens of this microscope. And he targeted the red indicator Varnum to one population of neurons and the green to another, and was able to acquire simultaneous spiking data from each. So that is allowing us now to study neuronal interactions in live animals in ways that was not previously feasible. Now, as I had described, voltage indicators offer the prospect not only of looking at neuronal spikes or action potentials, but also at the subthreshold facets of membrane activity, of membrane voltage dynamics. And so this is a topic that has long been in need of new mechanistic approaches. So brain oscillations, as I said, were known as early as the late 1920s, but exactly the mechanisms and cell contributions from the different neuron types that are required to induce oscillations of different kinds has been rather mysterious, because it's been hard to selectively record individual neuron types and to probe their subthreshold voltage oscillations. So to provide such a methodology, Jesse Marshall developed the following approach, which we call TEMPO, for transmembrane electrical measurements performed optically. And the schematic of his apparatus is shown here. He essentially used a reference fluorophore that could be engineered into the tissue to track hemodynamic effects and brain motion that would lead to artifacts in a voltage imaging experiment, as well as another channel for tracking the genetically encoded voltage indicator. And so these overall strategies to use the reference channel to correct for artifacts in the voltage imaging channel. And these channels were kept separate by fairly standard techniques in optics. They're both modulated at different frequencies, allowing one to demodulate in a way that leads to minimal crosstalk between the channels. And what Jesse showed was that this approach attains the physical limit on measurement sensitivity that is set by a photon shot noise. And so despite the fact that the animal can be running around, we can have hemodynamics, that with proper analysis of the data and use of the reference channel data, we can actually get down to the photon shot noise limit. So the virtues of measuring the voltage activity in this way allow us to do several things that conventional electrical field measurements, i.e. with electrodes, such as the electroencephalography or local field potential measurements in the brain, don't provide. And so first of all, we can target neuron types of our choice. We can identify these genetically or by virtue of their connectivity patterns. We also have the virtue of looking directly at transmembrane voltage. So in electrical measurements, there's a well-known set of artifacts coming from voltage conduction effects. In other words, one portion of the brain which you're not probing might undergo changes, for example, in its conductance that could affect your measurement, and you would not be able to correct for that. But here we're looking directly at the cell zone transmembrane voltage. We can track sparse cell types that are not dense in the tissue. And we also have some evidence that we actually have better sensitivity than the electrical measurements in non-laminar brain areas. And so overall, there are new possibilities for trying to dissect and find the mechanisms for brain oscillations cell type by cell type. So I'm going to give you some examples. So here in behaving mice, freely behaving mice, that are probed with this tempo fiber optic system, Jesse Marshall and Simon Haziza in the lab have been probing so-called theta and delta band oscillatory rhythms in the hippocampus of behaving mice. So when the mice are running, here you can see, as revealed by the tempo method, in a subset of cells that we targeted, the hippocampal pyramidal cells, a very nice theta oscillation within the 6 to 10 hertz band. But when the animal is more still, we see a rhythm in the so-called delta band here at lower frequencies. And what we found in the hippocampus, either with the optical approach is in good accord, it's in close agreement with what we found with an electrical measurement taken at the tip of the optical fiber. But of course, we can also use this optical method in brain areas where the electrical method does not work well. This slide shows you data from the hippocampus that there's a steady phase coherence between the optical measurement and the electrical local field potential measurements that we took at the fiber tip, giving credence to the validity of the optical data. And here is another example of theta oscillations that are evoked, this time in the visual cortex, when we showed the animal moving visual engratings. So stimuli of this kind that might be drifting across the monitor that the animal's looking at. And we found evoked theta rhythms in both excitatory populations of cortical neurons as well as inhibitory populations. But you can see there's a greater degree of reliability from trial to trial in the inhibitory neuronal population, whereas the excitatory cells respond, but perhaps less reliability, and both trial to trial and with regard to the timing relative to the stimulation, shown at time t equals 0. Now, one of the important things here is that we can target the indicator to different neuron types and dissect their different relationships to the electrical rhythm. So here's a case where we looked optically at the pyramidal cells, shown here in the orange trace, or a sparse population of cells called the parvalbumin interneurons, here in the green trace, and they had different phase relationships to the local field potential measured electrically. So now we can begin to see push-pull relationships, for example, between excitatory and inhibitory cells using optical voltage sensing. So we're very interested in dissecting the mechanistic contributions of the different neuron types. We've been able to target different cortical layers and even to look at higher frequencies. So here's an example of sensory evoked gamma rhythms. We were brushing the animal's whiskers and seeing an evoked oscillation here in the 20 to 80 hertz range, this time in the cortical pyramidal cells. So a lot of important new opportunities. And in fact, we've already been able to discover a novel rhythm with this approach in the medium spiny neurons of the basal ganglia. So when we targeted the indicator to the medium spiny neurons in the dorsal medium striation, we found one type of hyperpolarizing oscillation in these cells that had been reported, high voltage spindles illustrated here, but another type of oscillation which had not been previously reported, a hyperpolarizing rhythm that undergoes a fewer number of cycles than these high voltage spindles. So this was what makes us think we may have a greater sensitivity, at least in non-laminar brain areas, to oscillations that might not have been seen previously. And we've been applying this approach to different biological studies. So this is an example of a study that just came out, a collaboration with Vikas Sohal's lab at UCSF, in which we were comparing oscillations across the different hemispheres of the brain in this gamma range and seeing their possible engagement during different forms of learning, and in particular during rule shift learning. Now, in the final portion of the talk, I want to give you a sense of what we've been doing to improve the throughput of brain imaging experiments. And we've been focusing, in this case, on Drosophila, a very common model for the study of the invertebrate brain. And to further our high throughput approaches, Eric Totway-Ho and Joan Saval developed a robot that could track individual flies for the purpose of handling them and picking them up. So it uses machine vision to track the flies, and by suction can pick up an individual fly and move them about and manipulate them. And so there's an onboard camera here, infrared illumination. The flies they will not fly away if they're held in the dark. And this then allows us to manipulate flies, as illustrated here. So in this case, we instructed the robot to move all the flies from the left, put them on the right, and then it'll take all the flies from the right side and put them on the left side. This particular set of instructions is somewhat useless, but it gives you a sense of the flexibility and the versatility with which the robot can be programmed. Okay, so we can use the robot to provide a variety of different views of flies for morphological assessments using machine vision. This can be used for screening, for example. We can get precision measurements of the different body parts. And the robot can subject the fly to different illumination conditions that can highlight different parts of the body. In this case, for example, the wing. We can also use color machine vision to look at the morphology and to determine the fly's sex. And the fly's behavior can also be assessed with the robot. Here you can see the robot is holding a fly on top of this little spherical treadmill, and we can then monitor the animal's behavior in response to different odorants that we're delivering through this tube here. So in this next video, you can see a higher magnification example of this. We're delivering two different odors through these tubes, and the fly will respond in different ways to the different odors. And so in this way, we're moving towards automated assessment of fly behavior. Now, at the same time, we would like to be able to study the brain dynamics as we're watching the fly behavior. And so Supriyo Sinha, in collaboration with Tom Baer, developed laser microdissection techniques that were based on methods that had arisen in ophthalmic surgery. And so Supriyo used a 193 nanometer excimer laser to microdissect a portion of the fly's cuticle or exoskeleton. And here you can see some examples of this. By using different masks, we can dissect, say, a square area here, or in this case, a circular area. And the cuts are very precise, as you can see with these electron micrographs. They are a submicron precision. We even have the precision to ablate a single hair on the fly. So this is sort of the world's most precise haircut. Not something we need to do on a daily basis with a fly, but it gives you an example of how precise the dissection can be. Now, one of the virtues of this approach is that it has allowed us to create imaging windows in the cuticle that in turn enable us to follow individual neurons over extended time periods. So previous records in the fly imaging research field had been for periods of time on the order of 18 hours, maybe a day at most. And so it had been a challenge to follow individual neurons over extended time periods. But using our excimer laser-based microdissection, we could cut out an area of the cuticle, seal it with an optically transparent glue, and then come back again and again to the same neurons over time. So here's an example in which we have combined the optical voltage indicator targeted to individual neuron in the fly brain, and we've been able to follow these neurons routinely for at least 10 days, and in some cases up to 50 days. And so here you can see an example set of records of optical recorded spiking here in the fly brain using this microdissection-based approach. So I'm going to sum up now the different topics that I've shared with you, and I hope they give you a sense of some of the challenges that the neuroscience field has had to address and sort of where the field is at this point. So I started by talking about micro-optics and the ability to see deep in the brain. And as I said, nowadays researchers are using this and even in ventral portions of the mouse brain and applying it to other species. I discussed the miniature microscopes, which can be used to examine freely behaving mice. as well as other mammalian species. This provides the possibility of time-lapse tracking studies in which we can look at the evolution of ensemble neural codes, as I tried to illustrate in the hippocampus. More recently, we've been working on ways to image 2 or more brain areas. We took a look at the duopus and some of the preparations in microscopes for looking at 2 or more areas, including the crystal skull, this curved glass window, and the ability to track, in that case, many brain areas at once. And our efforts to look directly with optical methods at neuronal transmembrane voltage, which is giving us new ways to look at single action potentials with sub-millisecond precision, as well as to track the voltage rhythms in freely behaving animals. And finally, we've been trying to combine these optical approaches with machine vision-based robotic manipulation of the animals for high throughput experiments. So with that, I will stop, and I'm happy to take any questions if there's time.
Thanks for that fascinating talk. Do we have any questions? I ask that you introduce yourself. Masood, you're muted.
Thank you. No, I'm not muted anymore. You mentioned that you could identify neurons that are excitatory versus the ones that are inhibitory. How do you know that? Is it based on the shape of the neuron?
No, it's based on the genetics. We're genetically targeting them.
So how do you identify them under the microscope? Which one is which?
Well, for example, if we were to express the indicator in the excitatory neurons, then we would know that the neurons we were seeing that were emitting the fluorescence must be excitatory. So it's all based on the genetic promoter that we have selected during the genetic engineering. So meaning the answer to your question is the selection is not done optically. It's done during the genetic engineering phase of the experiment.
And all the synapses associated with one neuron are of the same type. So if the neuron is inhibitory, then when it connects to other neurons at various synapses, they're all doing the same thing, inhibition?
No. So let me clarify. So and maybe I should go back to those slides. Just give me 1 second. I will go back to that slide. So we are looking in our experiments at the postsynaptic elements. So in other words, we are looking at this. So let me see. Okay. So this is a dendrite from an individual cell, and these knobby structures are the dendritic spines. So these are all the postsynapses. So these are all excitatory synapses, which we know from other people's work showing that these spines only arise on excitatory synapses. And so these are all inputs to this cell. These are not the cell's outputs. So strictly speaking, we don't have a synapse by synapse record of where this is coming from. But in this particular case here in the hippocampus, we have a strong sense based on previous anatomical work that's been done at the aggregate level. But the images themselves do not show the presynaptic cell.
Okay, thanks.
Okay, I see someone called Ork Hangua has their hand raised. If you could introduce yourself before you ask.
Hi,
Yes.
thank you very much for a very nice presentation. I am Orhangwa, I'm a PhD student here. So I have several questions associated with the presentation. The first thing is that for the live mice brain imaging, what is the penetration depth? Like, are you imaging it from the surface or it's deeper than the surface?
Well, it depends on the area. So when we need to go deep, we use the microendoscopes. But if we're looking at more superficial areas, say like the neocortex or the cerebellar cortex, then we can just use the conventional objective lens, or one of these larger objective lenses, like with the 16-beam system.
So like how deep it's supposed to go, like deepest point, for example? Like is it like millimeter or micrometer level?
Well, let me go back to that slide so we can take a look at it. So you know, so each tick here on the scale is 1 millimeter. So you can see that we can have endoscopes, you know, that can be really quite long, and this, you know, with the say, you know, roughly 1 centimeter endoscope, you can go to the ventral portion of the mouse brain. Now, without the endoscope, you would be limited to hundreds of microns from the surface of the tissue, and so that, you know, which I illustrated at the beginning of the talk. So here you can see some of the penetration depths of the conventional microscopy techniques with these colored bands indicating the depth. So it would be sub-millimeter unless you use the endoscope typically.
So second question is that what I noticed is the frame rate of the imaging seems like about like 20 hertz or so, if I understood correctly, for the miniature microscopy. So what limits that frame rate? And could it be like faster, or being faster would be helpful?
Oh, it is faster nowadays. So this is just based on the CMOS image sensor that we select. And, so depending which one you choose and which mode you run it, it can go quite fast, and they can go up to 60 hertz now.
So the third question is that what could be the ideal laser parameters in terms of the central wavelength, specifically for those multiphoton imaging microscopes, like and then pulse duration or peak power or something like that?
Yeah, so the most common laser that people use is typically the Ti-sapphire laser, and that's because there's multiple parameters that are attractive. So first of all, people like the tunability, so you can actually excite different dyes or fluorophores with different absorption spectra. Of course, one wants to have short pulses to improve the excitation efficiency. Repetition rate is important. If the repetition rate is too slow, this will limit the imaging frame rate. On the other hand, if the repetition rate is too high, this will limit the amount of energy per pulse and will reduce the excitation efficiency. So in our field, we do not tend to use repetition rates as high as the gigahertz, which you might see, for example, in telecom. It's nice to have repetition rates in the order of, say, sub-megahertz to up to 100 megahertz would be the typical range. Now, there's, of course, a limit on the total power that we can apply to the brain due to reasons of brain heating, photobleaching, and phototoxicity. So there's improvement to be made. People are exploring 3-photon imaging in the field, and there are, of course, different wavelength requirements for 3-photon imaging, as well as the fact that it's typically good to have amplification of the laser pulses. But for conventional imaging, parameters might be 80 megahertz repetition rate, say 800 to 850 nanometers wavelength of excitation would be, what's typical in an experiment.
I see. So, like, how about the peak power? Like, how much peak power is desirable, like sufficient, for example?
Typically fractions of a nanojoule.
I see. So if it's like very short, then it's about like kilowatt peak power range. Okay. Thank you very much. That's all I have.
Thanks. Do we have other questions? Oh, let's do it again. You're muted.
Can you control the behavior of the animal by exciting specific regions of the brain, or are you simply monitoring the reaction of the response of the neurons to the specific behavior?
We do both. So, I mean, we certainly historically came at this from the perspective of imaging, as I, mostly talked about. I did mention optogenetics briefly, and that is sort of a field in its own right and has been, largely initiated by my Stanford colleague, Carl Deisseroth, who developed the use of light-gated ion channels for manipulating the neuronal transmembrane potential. And so you can target those proteins as well, as we were discussing earlier for the fluorophores, to particular neuron types to either excite or inhibit neuronal activity of defined cell types.
And how about correlations between the two hemispheres? Do you see the same behavior or related behavior on the two hemispheres for a given event?
It really depends on the behavior. There are correlations, but of course the two hemispheres are not behaving identically. But, as I mentioned, it's actually rather new to be imaging across these larger areas. And so, that's, in some ways, that's an area of investigation. But certainly it's known, if you go into the human brain, from what one can tell, say, with fMRI, that there's quite a bit of lateralization.
But you can confirm what has been suggested in the past, that, for example, the right eye is connected to the left brain and vice versa.
Yes, and there's also a bilateral region as well. Yeah.
Okay, thank you.
Do we have other questions?
Hi, I'm Haijiang from neuroscience, and it's a really fantastic talk. Can you comment a little bit about how the miniaturized microendoscope will work with the voltage sensor dye, and also can you comment a little bit about the recent development of the voltage sensor dye working with this imaging tools?
Yes. So, okay. So first of all, we have not yet combined the voltage sensitive indicators with the microendoscopes. That is something we would like to do, but it has not yet been done. And, I think as you're implying, the voltage imaging brings its own set of challenges to microscopy. So to track spikes, it's nice to have frame rates on the order of a kilohertz. So you need to have scientific grade CMOS cameras typically that can run at that rate with low levels of read noise. The conventional 2-photon microscope does not have frame acquisition rates at that level. So there's a whole set of microscopy challenges that are going to have to be addressed for those voltage imaging experiments. And those challenges will also be distinct whether you're interested in imaging spikes or the subthreshold voltage oscillations. So I think you saw in the talk the tempo instrument, which was really targeted towards looking at the subthreshold oscillations, not the spikes.
All right, thank you.
Okay, do we have other questions? So you showed a lot of imaging work. Earlier we'd been talking about these optical resonators that I use. Is there any room to sort of combine these two technologies from your point of view, where you're getting this neuronal firing and maybe doing some like biomarker detection?
Well, people are, of course, very interested in biomarkers, and biomarkers are important in human neuroscience as potential predictors for various conditions. There's also interest in trying to understand what might be the more microscopic basis for some of those biomarkers, and that's where some of our imaging techniques might come into play. With regard to the first part of your question, with regard to the optical resonators, everything we're doing is obviously sort of an intact tissue, if you will, in live animals. And I would defer to you with regard to the resonators, but what I sort of imagine is that given the sensitivity that you have, that you would probably need more controlled environments.
Well, we do have some selectivities, but I would, so do you ever so everything's in the tissue, so I guess you guys don't extract any like fluid or anything while you're doing the experiments? I guess obviously not.
We can, but in many cases we're doing these long-term imaging experiments, and in fact the tissue has been sealed.
Okay, just some thoughts. Okay, so do we have any other questions? If not, I'd like to switch it over to the meet and greet portion of the event, which is basically when students can introduce themselves and ask questions that are not necessarily related to the talk. Do we have any students that want to go? Otherwise, I have some icebreaker questions that I typically ask.
One question, and maybe this is just my lack of knowledge of the neuroscience part of everything. My name's Sarah. I'm a master's student at the Optical Science College, and I'm wondering, because you're implanting these microendoscopes, is there any tissue damage caused by the implanting of them? And if so, what's the long-term effects of that?
Yeah, there is some damage. So, obviously the tissue and the glass probe cannot reside in the same portion of space at the same time. So there's unquestionably, we are displacing some of the tissue. moreover, when you insert a foreign body into the brain, you will see what's often called a glial scar form around that tissue. So this is well known from electrode studies. If you put an electrode into the brain and permanently implant it, you will see a sort of a scar region of about tens of microns in contact with the foreign object. And we see that at the tissue that is contacting the microendoscope. Now, after a week or two from the initial implantation, that zone of tissue has become stable, and we can inspect that, say, using conventional histopathology. And so what our imaging studies are doing is actually looking through that zone of tissue. So we have a working distance that is beyond that region. And so we're looking at very stable tissue portions. And as you could see, for example, from our imaging studies of adult dendrites, they're not changing. There's no sign of neuronal degeneration or anything like that over long periods of time. And in fact, in some animals, we've been able to track them for over a year. So I think our perspective on this is, yes, there is a perturbation. We are looking beyond the perturbed tissue. We do not see any changes in animal behavior based on this, so there's no gross or macroscopic effects on this. And so our perspective is that it remains a useful experimental tool. To put this in context, I think it's worth mentioning that our surgeries are far more delicate and precise than what takes place in human brain surgery. So if you were to, watch a human brain surgery in process, I think you would see things that were, far more perturbative than what we are doing in the mouse brain. And it's well known, for example, that objects can be placed in the human brain without overt long-term effects. So, there's an endoscope that gets inserted into the human brain that can be millimeters in diameter. It can go through the interior cingulate without overt effects. And as you know, Parkinson patients are being implanted with neurostimulators. That's invasive brain surgery. So I think, is there a perturbation? Yes. Does this remain a useful experimental tool? Yes.
Thank you.
Thank you. Thanks for the question.
I see Uptal has his hand raised. You're muted.
Hello, thank you for the wonderful talk. I just had a curious question. How do you take care of all the mice?
So typically biology departments will have individuals whose chief task is mouse husbandry.
Okay. So are there any rules you guys have to follow to make sure there's no violation of animal organizations, like to take care of their well-being or something? You don't cross the line of...
Yes, all the experiments are carefully reviewed by a panel before any experimentation takes place. And this panel includes scientists and veterinarians, and typically at least a clergy and a member of the local community. So they are carefully vetted, and I'm confident that Arizona does it the same way. And in fact, there's also a national accreditation process that takes place that is critical for publishing. So there's very strict standards and oversight in all animal experiments that would be, standard and typical across all major research institutes throughout the country.
Is there any special training students undergo before they enter the lab?
Well, we would consider being in the lab and doing research part of the training. Now, perhaps in light of your previous question, maybe what you meant is there a training that takes place before they would say work with the animals?
Yes,
as opposed to the lasers. So absolutely, they get training, and they have to go through particular courses to start.
Okay, thank you.
And I see Orkanga has her hand raised again.
Yeah, so I work with fiber lasers, and then I actually built one laser that is tunable from 700 to 900 nanometer. It's all fiber source, and then it can give few nanojoule pulse energy. And have you tried to use fiber lasers instead of tie sapphire? Because I was curious about because tie sapphires can get to millijoule levels. So I thought you use plenty high pulse energy, but it seems like you use very small pulse energy too. So
yeah, people have used fiber lasers. I'd say the most common has been either ytterbium or neodymium. But yeah, people have used them. Yeah, for sure.
I see. So there is like no really specific, it's just because it's easier to get, that's why you use tie sapphire later?
Yeah, I think it's mostly a matter of convenience, but people are branching out. So there's, I mean, I think probably the most common would be ytterbium, but yeah, people are branching out for sure.
I see, because I built like fiber OPOs using photonic crystal fibers, and from ytterbium, then we could get like exactly the wavelengths that you seem to prefer. I think like for brain calcium, it's maybe around like 900 is desirable, right? So
yeah, no,
900 nanometers.
These sources are definitely applicable. I think, you know, people in our field and especially the biologists are, you know, their focus in their research is on the question about the brain, right? And so they're less interested in building their own laser and more interested in having the laser be purchased commercially and be reliable for multiple years, right? So the laser is viewed more as a tool in this field. And then our effort, you know, would be using this well-chosen light source as the basis for our microscopy. But you are absolutely correct in saying that, you know, there's been great innovation in ultra-fast light sources and that they are indeed applicable to multiphoton imaging. No question about it.
All right, thank you.
Masood?
You didn't say much about the glial cell. Can you monitor those as well? What's the difference between their behavior and the ordinary neurons?
Well, that's a great question. The glial cells, i.e. the non-neuronal cells in the brain, are actually the majority. And in, say, the last 2 decades, they have gained increased attention in the research field. Historically, people put a lot of attention on the neuron because those are the cells that are very obviously electrically responsive. So in particular, neurons have sodium channels and can fire action potentials, giving them the basis for millisecond scale communication. So, you know, for example, my visual cortex is here at the back of my head, is 2 synapses away from the retina, and those signals can traverse my brain in milliseconds. And so that's due to the neuronal action potential. And so, you know, you can imagine this is what captivated neuroscientists as the basis for neuronal communication and information processing. Nevertheless, as time has gone by, there's been increased appreciation that glial cells and the various subtypes thereof may be doing very important jobs in the brain. And so there has been a lot of research more recently on how they may be helping to regulate cerebral blood flow in response to neuronal activity, modifying synapses, i.e. synaptic plasticity. And in fact, it is known but understudied that glia often have electrical dynamics of their own, but just not fast dynamics. They have slow potentials, but they remain highly understudied. So, but to answer your question, one can look at the dynamics of glia, and in particular, a subclass known as astrocytes, people have examined with calcium imaging with the techniques that are similar to what I showed. In many ways, they're easier to look at than neurons because they lack the fast dynamics. So there's time to get more photons.
What are the typical sizes in terms of diameter and length, number of axons, etc.?
Of a neuron?
Glial cells.
Well, they don't have axons. They don't have axons.
Dendrites?
Sorry?
How about
well, they wouldn't be called dendrites. They would be processes. But the astrocytes in particular can be quite large, actually. So they could be, say, sub-millimeter, hundreds of microns for sure. And they can have a great number of parameters. And they do have a different morphology than the neurons. They can have quite a complicated arborization. Whereas other types like microglia would appear quite differently.
Thanks.
Do we have any other questions?
I have a quick question. I apologize for being late. Professor Schnitzer, I was curious, as you've been, I guess, further developing the understanding of the brain in general, and collecting all this data about how we are trying to understand how the different areas of the brain communicate, what has been your go-to approach for analyzing the macro scale of all the activity simultaneously? Is it like, or sorry,
Well, I was going to say, it depends a little bit on the experiment, right? I mean, it depends on the particular question or hypothesis that we've generated. But by and large, you know, we're interested in the relationship between what the animal is doing and what the neurons are doing. And so we try to discover that relationship in various ways. Say, first starting at the analyses that would allow us to focus on single cells, but then analyses that would allow us to focus on the population of cells. So particularly at that second level, we often find ourselves now actually using machine learning techniques to make, you know, statistical inferences about what group activity amongst the cells will tell us about the activity of the mouse.
So when you are using the machine learning techniques, I guess, are you analytically representing them, or are you setting, like, are you perhaps representing each neuron as a cell, or is it more like over the cluster of electrical activity you recorded, feed that as input into a network for analyses?
It depends on the analysis. So in many analyses, you're simply using them as methods of statistical learning, basically just learning patterns. So for example, when I don't know if you were present for the portion of the talk where I was discussing the hippocampal representation of space, but there we were taking data as input, the neuronal dynamics from the hippocampus, and making a prediction about where the mouse was in its track. And so we use a Bayesian decoder for that, a very simple technique in statistical learning. There's no attempt to represent the actual dynamics that would be internal. It's simply a matter of predicting, in that case, you know, the animal's most likely location given the observation in the neurons that you just saw. Now, there are efforts to try to actually model the activity, in which case, you know, you might try to model the actual neurons that you saw, or just representative neurons, right? So you could try to get the gist of what was going on. You wouldn't necessarily have to model each and every cell that you saw. You might just be trying to explain the data, say with a theory. But by and large, our data analysis techniques are making no attempt to represent the particular neurons per se, but to really find, you know, relationships in the statistical patterns of neuronal activity to animal activity.
Have you seen any indication of chaotic behavior, like feed-forward positive feedback that creates a storm in the brain?
We haven't looked at that, but I think if you look in the literature, you'll see some reports that might be consistent with that, and theorists have looked at that also.
I guess when I think of neural networks as like an analog to not necessarily how the brain works, but as they like to market it, I think we're also doing a very difficult job of visually representing the different activity. I mean, we're showing a cross-sectional view flattened into 2D space. And I guess I'm curious, something much more complex like the human brain with so much more neurons and different types of action potentials, or maybe not different types, but different types of electrical activity going on. I'm just curious how biologists or neuroscientists really, it doesn't seem like it's a similar problem, like in terms of trying to model everything going on a spatial level. I was just curious, as you're imaging and collecting even more data and analyzing it, how is it that we're spatially representing everything going on in adjacent neighborhoods of the brain?
I'm not sure I entirely understand the question. I mean, we try to maintain the coordinates where we try to maintain the coordinates from where we took the data. So we do know something about the relative anatomic locations of the cells, and we try to come back, you know, to the same brain area in individual mouse or across different mice. So I'm not sure that entirely answers you. I mean, people who do brain mapping studies in humans, they do have techniques for, say, you know, if my brain was part of an imaging study and your brain was, you know, for sort of morphing and mapping the corresponding areas on top of each other, and there are image analysis techniques for doing that. I'm not entirely sure I'm addressing your question, but
I guess if you were imaging multiple regions of the brain simultaneously, and you wanted to see the data side by side on the same, I guess, spatial plot.
Well, that's, I mean, that's, you know, that's not hard. We just put the two graphs next to each other.
Oh, right. I mean more like, but the coordinates actually correspond to like grounded XYZ information inside the brain.
Yeah, I thought I showed you some of that in the talk, right? I had these two movies. And then so that preserves the coordinate systems.
Gotcha. I was, okay, cool. I'm trying to, yeah, okay. I think that does answer my question.
Yeah, I mean, if you look behind me, right, you can imagine we could get one field of view here, another one here, and you could show those simultaneously and preserve your information about where the activity was coming from.
Gotcha. Looks very cool, by the way.
Yeah, thanks.
I see Uktal has his hand up.
Yeah. So how similar is the mouse brain to the human brain? Are we able to extend the results obtained on a mouse to the human?
Well, it's a good question. There are different answers to this question. At some level we don't know yet, right? Now, another way to look at this is that, of course, there has to be some difference because our behavior is very different. We're learning more about the differences in the types of cells that comprise the human brain, that comprise the mouse brain. This has been one of the foci of the NIH Brain Initiative. So there's been a cataloging of the different neuron types across the mouse brain. That's been published, at least portions of the data, and the human brain is coming, I believe. So we're learning, more about the unique features of the human brain. Having a parts list is not sufficient to describe all the differences, of course, because you need to know the projection patterns of those neurons and their higher order statistical correlations. There's also going to be microscopic differences, say, for example, in terms of levels of RNA and protein expression. So you're going to find a lot of commonalities. And this, of course, is why we study the mouse, because we believe we are going to learn something about the human brain. But there are going to be important differences. And that actually is one of the motivations that researchers have is for studying the primate brain. There may be some things that are not well captured by mice, right? So it really depends on the question. The subcortical areas tend to be more ancient from an evolutionary perspective. So basic functions such as, breathing, eating, drinking, these functions are likely to have great commonality between different mammalian species with some important differences. But when you look at the neocortex, which has evolved more recently in evolutionary history and underwent a great expansion in the mammalian brain, these areas are likely to be some of the sites of, more noticeable differences.
Okay, thank you.
Any other questions? So if not, I'd like to just end on one final question. What's the best piece of advice you have for graduate students?
Well, find something that you're passionate about and study it.
So
I have one more question. I guess one I come from more of an information theory perspective, and I know a lot of Shannon theory has been not bandwagoned in various fields, but I do think that in neuroscience there is a very dense amount of information here, and I guess I'm trying to imagine as we actually glean more information about what's going on inside the brain, how do you think analytically, not necessarily the hardware technology, but analytical visualizations and other technologies will develop as we try to get a bigger picture of all this information going on?
So I'm not really sure if your question is asking me about visualization technique or you're asking me about the applicability of engineering theories to the study of the brain. Which are you talking about tools that are come out of neuroscience or that should be applied to neuroscience? I'm not sure I fully understand your question.
I guess it's a little both. When I think about when scientists are looking at neural networks and they're looking at they're looking at this, as Professor Masood mentioned earlier, kind of a cluster of all this neuron activations in a neural network. And I see how we represent it currently in mathematics or different fields as like a flat cross-sectional representation. And I'm just trying to, we're mapping I guess in the CS description of things, we're mapping it into this higher dimensional space. And often we lose track of what's going on in these higher dimensions. And the brain, I guess, I think of it as such a very complex like a graph structure. And with our 2D representations, often like my analysis is limited to when I'm looking at the big picture of what's happening. And so I'm trying to imagine as new analytical techniques get developed. Like I always was a big proponent of holography because it's just like, you know, all 3 dimensions of information stored in flatland here. I'm just trying to imagine, like, we're only going to learn more about the brain, and I'm trying to imagine how we are going to develop, how technology will develop, how you think technology will develop to understand this bigger picture as we continue to learn more.
Yeah, well, I guess I can try to answer that in 2 ways. So, so first, you know, people apply various analytic tools when they have appropriate questions to address. Mm-hmm. So, you know, of course, you know, analysis tools have to be matched to questions. There's, you know, it's a little bit like asking, you know, are you in favor of a hammer or do you prefer screwdrivers? Well, you know, it depends if you have a nail or a screw, right? So it's hard to talk about the relative merits of a tool, you know, without looking at the problem that you're trying to face. Mm-hmm. Now, having said that, neuroscientists are very interested these days in neural nets and machine learning because they are finding increasing usage in these approaches for addressing a variety of questions. So, you know, as we were talking about earlier, some of them are with modeling networks, right? Can you capture the behavior with models of artificial networks? But some of them are just for data analysis, right? So there's no attempt to model. But you know, I think neuroscientists are increasingly appreciating that approaches that have been developed in computer science, electrical engineering, statistics are going to be relevant. And neuroscience programs have become far more interdisciplinary than they used to be. So if you go back not that many decades, the word neuroscience was not in common parlance. And people used to be trained in neuroanatomy or neurology, but neuroscience itself is even a more modern word. And it connotes an interdisciplinarity that some of the older specialties didn't have. So for example, you know, like I said, you used to have neuroanatomists, and they, you know, might or might not have expertise in the physiology, in the information processing. Whereas now I think there's increased recognition that we need to be teaching students about a variety of different pertinent disciplines. We need to be taking students that might have had undergraduate training in other fields. You're seeing, you know, new domains such as neuroengineering, neurotechnology crop up. Engineering departments are hiring in these areas, right? You're seeing movement in industry. You know, Elon Musk has a company, right? Neuralink. So you're seeing a lot of interest in overlap with other fields and interdisciplinary flavors of neuroscience. So I'm not sure I entirely answered your question, but I think you're going to see sort of a lot of activity and increasing interest from the engineering community.
No, I think you did. I guess I'm curious, as you pointed out, every model is specific to every question. And, but now when I think about neural networks, the computational computer science neural networks, I see them using the same diagram, you know, for different tasks or techniques and sometimes I worry that the diagram, there could be better representations of relating to whatever task they're doing, or even a more generalized representation, no matter the task. And so while you pointed out for certain modeling techniques, certain image tasks, there's certain modeling techniques. I was curious if there's a nice general model that neuroscientists like to relate to or point to that allows people to cross between the different research topics or investigations. But I think you pointed out that there's
there is no unified model of the brain yet, for sure.
Yeah.
And so, you know, we use different models appropriate to different experiments and different questions.
I see. Cool. Well, thank you so much.
Thank you.
Makes me think about more questions. Yeah, thank you.
So I just want to thank Professor Schnitzer again for taking the time to talk to us, and it was like such a fantastic talk, and we really appreciate you being here today. And
thanks for the invitation and for all the great questions. I mean, the questions are, I hope, an indicator of interest in the field. So I appreciate the inquisitiveness.
I think that's the most intensive question session I've ever seen.
Great.
Great. So thanks, and everyone have a great evening.
Thank you
Okay,
very much.
Bye.
thanks again. Take care.
Bye-bye.